All right, we're in the home stretch here, and now we're getting to look at some of the really interesting data here. So this is where we'll actually get some answers to our original research question, which was whether the bacterial communities are different in male and female garter snakes. So since we've now finished running core metrics, we have these different options that are available for us to view. We're going to start here with the beta diversity. So click on that one. And there are several options here. For us, let's just look at this Bray-Curtis distance and click on that emperor plot, and it'll load a plot for us. And we can customize this. So for color, under select a color category here, select sex. So what that'll do is now we can easily differentiate males versus females. That's what we want because we are interested in whether there are male versus female differences. And then you don't have to, but I don't like reading graphs with a black background. So if you click axes, you can change the background to white and the lines to black. And these are three-dimensional plots, so you can actually click them and move them around, which is fun. But overall, what we're looking at here is whether these different samples are clustering in a three-dimensional space. So the way to read these types of plots is basically each data point represents a sample, and each sample's position in three-dimensional space is based on the abundance and relative amount of all of those different bacterial species that are present in each one of those samples. So this is basically a way to condense all of that information from all of the different bacterial taxa and condense it into one single data point in three-dimensional space. So really what we're interested in when we look at these types of beta diversity plots is whether there are clustering of samples in three-dimensional space and whether that clustering is matching or representative of what we thought might be happening in terms of sample differences. So here we have, if we go back to color, so again, all the red ones are female, all the blue ones are male. And we can see that in general, especially on this axis 1 here, we see separation where we see all of the female samples here on the left and all the male samples here on the right. And overall, the male samples seem to be clustering together and all the female samples seem to be clustering together. So that's really interesting, and that leads us to believe that there are probably some differences in the microbiome of the male and female garter snakes, which is cool because when you start experiments, you kind of have expectations and hopes, but it's cool to see that there is evidence at this point to support our hypothesis that there were differences between male and female garter snakes. So I'm not asking you to save this plot for our lab report, but you can if you want. So if you right click, you can save the image as a high resolution PNG. Okay, so that's the beta diversity. So from this step, we think that there probably are some bacterial differences between males and females. And what we'll do next is look at that in more detail by clicking this taxonomic diversity. And what you want to do here in these results is click on bar plots. And we will choose taxonomic level 6, which means we're going to differentiate the different bacteria by genus. And we are going to sort our samples by sample. And if we scroll down now, what we have is a taxonomic diversity bar plot that is showing us for each of our samples. Now we have all the female samples on the left, all the male samples on the right, and our y-axis is relative frequency. So it's showing us for each of these samples roughly how common or how abundant each of these different bacterial taxa were in each of those samples. The right-hand side here is a color-coded legend that corresponds to each of the different bacterial genera that were present in these different samples. These plots are interactive, so if you thought if you hovered over one of these, it would pop up for you. Maybe not. Okay, but what you can do is with this legend on the right, you can click and play around with which of these different ones you want to show. So I clicked this one right here, Eubacterium, and now in our graph, those are the only genera that are being shown. If you decide you don't want to do that, you can re-click it again, and it'll restore that original plot. So again, remember our original research question was whether there are differences in male and female garter snakes. So what I want you to do is play around with this. So click around and try to identify bacteria that are only present in female snakes or only present in male snakes. So if we click this one, for instance, this is Methylobacteriaceae, and we scroll down, we see that this is present in 1, 2, 3, 4, 5, 6 of our female snakes, but none of the male snakes. So that's really interesting, and this indicates that this could be playing a role in the hormonal mating for the snakes or something like that. But overall, our evidence from here suggests that that bacterial species is only present in the females and not the males. So that's really what we're interested in here. So what I want you to do for the lab is click around and identify at least 2 bacterial genera that are present only in females. And when I say only in females, present in at least 3 females. So if it's only present in 1 female, that might just be sampling error. So pick 2 bacterial genera that are present in 3 or more females and only in females, and 2 or more bacterial genera that are present only in 3 or more males. So like I just showed here, here's one for you, that one's only present in females, and you can click around. So if I wanted to add in an additional one, so now I've clicked both this pink and blue one, and actually both of those look like they are only present in females. So that's the way for you to kind of build this customizable plot. If you click one and decide you don't want it, so that one is present in females and males, we don't want that one, you can click it again and it'll go away and return to just the 2 that you had clicked before. So play around with that and identify some differences between males and females. And then when you have a plot that you are satisfied with, that you want to put in your lab report, scroll back up to this download section here. Click SVG bars, that will download the bar graph, and SVG legend will download the legend for you. And then you can put that information in your lab report.